Okay, everybody, good to see you all again. I am fulfilling my promise to the attendees of the workshop that we ran last week at the Society for Psychophysiological Research. I gave this presentation to start the day before we really got into the tutorials about analyzing neural oscillations and aperiodic activity. And then a bunch of people asked if I had a recording of the presentation. We didn't record it, so I am now recording it for you back here in sunny San Diego. So I guess get ready to speed this up a little bit and play at 1.25 or 5 speed because this is a pretty long presentation, but it will guide you through the way that we've been thinking about the neural oscillations and what we've been calling aperiodic activity and what the physiological and functional relevance of that aperiodic activity could be. All right, so to begin, I want to thank everybody in the lab, first of all. A lot of these folks were critical for building the tutorials, the Google Colab notebooks that we're going to be sharing and so on. Andrew Bender, Blanca Burgos-Martin, Kirina Van Engen, Ryan Hammonds, Ina Kosick, Trevor McPherson, MJ Preston, and Sydney Smith. And so these are the folks that built a lot of the stuff that you're going to be looking at, the tutorials and so on. They come from the neurosciences, cog sci, and data science PhD programs. And because it's UC San Diego, have to have the it's a requirement for us to have the beach picture. It's a really great lab. I've been having a lot of fun with them. I was so happy to be able to go with them to Vancouver for the SPR conference. That was our first real big group conference together in a couple of years. So that was fantastic. So oscillations, what are we going to be talking about today? I love oscillations. I'm a big fan of the idea that oscillations have a functional role to play in interregional information flow between brain regions, cognitive functioning, disease. So what's the problem then? Oscillations are exciting. They're interesting. You stick an electrode into just about anything's brain, and you will see these rhythmic fluctuations in voltage that are these oscillations. They're pretty ubiquitous. We see them across many, many species, many brain areas. So why is there a problem? What are we talking about here? The first issue that I began to run into during my early days of science was that oscillations are correlated with just about everything. There's tens of thousands of papers talking about neural oscillations, maybe even hundreds of thousands. But if you go into PubMed and you type your favorite oscillation frequency band, so you have delta, theta, alpha, beta, gamma, high gamma, sigma, ripple, what have you, and your favorite psychological cognitive construct, maybe the disease model that you work with, what have you, you type those two things together, oscillation frequency band name, and some kind of function or disease, you will find papers that talk about the two together. And if everything is related to everything, what does that mean? What are we to make of that in terms of the neuroscience behind these things? And so I got stuck on this problem for a long time and ultimately led me down this years-long rabbit hole, methodological, theoretical, computational, physiological rabbit hole of what is a neural oscillation? What do we actually mean when we say a neural oscillation? And so broadly speaking, there are two tactics that we take in my lab when we're talking about oscillations, and that is we can't talk about them without first understanding, one, where do oscillations come from? And two, how are oscillations traditionally measured? I'm not going to spend a lot of time and focus on point one here, because the point of this workshop that we ran at SPR was to teach people the methods that we and others have really been developing in the last couple of years to really break through and give new opportunities for oscillations research. And that comes from understanding how they're traditionally measured. I will very briefly touch on some of the experiments the lab is doing on point number one here to begin with, which is this idea of where do oscillations come from? The fancy term for this would be oscillogenesis. Where do they arise from? And so one way that we've taken to doing this, thinking about it in terms of their physiology, which is, I believe, where we should be starting. Yuri Bujaki, Anastasio, and Cook have a great paper from 2012, Nature Reviews Neuroscience, looking at the origin of the local field potential EEG and so on. Gaute Einevol has done a lot of really fantastic work on the computational modeling side related to this as well. And so very largely, broadly speaking, when we're talking about oscillations, what we're talking about is some mass action in the local field potential, or by extension, EEG. That is, if we have a penetrating electrode into a single neuron that's recording intracellularly, then what we're picking up is the output from that neuron. We're picking up the spiking activity of that neuron. If we have an extracellular penetrating electrode, or even a surface electrode on the cortical surface, such as like in the LFP or electrocorticography, or then by extension, non-invasive EEG, we're not picking up the spikes, we're picking up the postsynaptic and transmembrane currents that are coming into that region. Tens of thousands, hundreds of thousands of these currents that are all converging on these regions, excitatory currents, inhibitory currents, neuromodulatory currents, all of these different currents. And that is what we're picking up. And so in terms of the understanding oscillogenesis, where do these things come from? One tactic that we've taken in the lab is by working with collaborators Alison Motri. This is a paper we had in 2019 with Clever Trujillo, Richard Gao, former PhD student in my lab, and Pri Nagres, looking at cortical organoids. So if you're not familiar with this model, cortical organoids are, I'm not a cell biologist, so I'm just going to wave my hands and say that they're cell biology magic. But it's a model system that is increasingly being used, whereby you take skin samples from people, usually like a cheek swab or something like that. You then, through the magic of cell biology, coax those skin cells into becoming pluripotent stem cells, and then you can grow them forward as neural tissue. And they form these cortical organoids. These are about 4 millimeters in diameter, approximately 100,000 neurons. They don't have microglia, they don't have vasculature. They're about the size of a bee's brain. They're very, very small. But these cortical organoids, over time, in the right medium, begin to laminate into what looks like laminated neocortex-like laminations, which is pretty fascinating and interesting in and of itself. If you record electrophysiology from them, what you see very early on, these are plots here of network spikes, which is if you time lock your electrophysiological analyses to the onset of a spike, and then you look at all of the spikes in the network. What is the average firing rate, the number of spikes per unit time of all of the spikes that you're recording from simultaneously in these cortical organoids? You can see that when one neuron spikes, they all spike within a very short time window, and then quickly those spikes abate. And they actually go silent for tens of seconds, and then you see another big network spike, and they go silent for tens of seconds. And so when this collaboration first really started between my lab and Alison Motri's group here, it was sort of arose from this, where Alison Motri was making the argument that these cortical organoids give us an unparalleled opportunity to study very early human neurodevelopment. This is something that we just really can't do. We don't have ways of studying neurodevelopment at this early of a stage. And so this, to me, is very exciting, but as a model for healthy functioning brains, this doesn't look like that. The human brain doesn't have everything spike all at once and then go silent for tens of seconds. And so I began to ask, at what point do the spiking rasters of these cortical organoids begin to look a little bit more normal, like healthy functioning cortex? And so Alison Motri's group began to continue developing these organoids for longer and longer time periods. And what they found is that somewhere around the 4-month mark, you see that when a network spike is initiated, you actually begin to see secondary and tertiary ripples. That is, everything spikes, goes quiet for half a second, spikes again, abates, spikes again. That looks a lot like an oscillation. And if you look at the LFP, relative to the network spike at 2 months, you don't really see much. Same at 4 months, but at 6 months, you see the network spike and then a lot of subsequent ringing in the spiking activity around that. And in the LFP, you see something very similar. And by 8 months, you begin to get this heterogeneity of ringing frequencies. And the LFP starts to also show an increase in complexity and the number of different kinds of frequencies that are represented in these very tiny networks. And so to us, this is very exciting. because it was the first observation that you get the spontaneous oscillogenesis in these cortical organoids. And of course, then the big question for us in our ongoing collaboration with Motri's group is, what's happening between 4 and 6 months that allows for the oscillations to emerge? That is, what's happening whereby the oscillations are initiated. So early in neurodevelopment, GABA, which is the primary inhibitory neurotransmitter in the human neocortex, is actually excitatory as well. And at some point during development, it undergoes a sign flip and becomes inhibitory. Also, in early neurodevelopment, you have excess connections. There's a lot of connectivity that happen before the pruning back of axonal connections between things. So everything is hyper-connected, over-connected. And so maybe there's some aspect of the GABAergic sign flip, where in order for these oscillations to emerge, you need these excitation inhibition dynamics. It's unclear, but this is part of our ongoing collaboration. So that's, to me, very exciting work that we're doing, trying to understand physiologically at the lowest levels where are oscillations coming from. But what the rest of this presentation is about, what the workshop at SPR is focused on, was how are oscillations traditionally measured? And why is this something that I care about? Because it should be pretty straightforward. So this is my lab is very big on GIFs, like animations that help you visualize some complex ideas. So I'll be showing a number of GIFs here from this first one from former PhD student in my lab, Tom Donahue, and then quite a number from Natalie Schwarnikov, who was a former postdoc in the lab, who did some fantastic work on creating a lot of these animations. So this one here from Tom is trying to illustrate the idea of the Fourier transform, which is just one of those mathematical marvels, just these fundamental truths that to me is amazing that anybody figured out hundreds of years ago, which is anything that can be represented as a time-varying function can be mathematically perfectly captured as a sum of sinusoids of varying amplitudes and frequencies, or varying power and frequencies, and technically also starting phases. And so here is a time series that you might record, say, from an EEG or LFP from somebody there on the bottom left. And you can begin by adding different oscillations of increasing frequency, which also happen to have decreasing power with increasing frequency. This top plot is also showing the starting phase of each one of these oscillations. So you can see here, this is like very, very slow one here, begins sort of closer to the trough. And some of them here, like the CLO, begins closer to the peak. And as you add more and more of these, then you get eventually a perfect mathematical capturing of the original time domain signal. So you can move back and forth between the frequency domain and the time domain without any loss of information. And so when we're talking about neural oscillations, the standard method for measuring them is using the Fourier transform, or very closely related mathematical concepts, to look at the signal in what's called the frequency domain. So our time domain neural signal here can be represented or captured by a sum of sine waves of varying amplitudes and frequencies. And this is what neural data tends to look like here on the right. So you can see here, this is a log frequency here on the x-axis by log power on the y-axis, starting at 1 hertz, 10 hertz, 100 hertz. And you can see the very lowest frequencies here have exponentially higher power than the highest frequencies. This is log y-axis as well. So we're talking about a huge exponential orders of magnitude difference in power between the lowest and highest frequencies. And right here, in this case, right around 8 hertz, you have a bump in power in a narrow frequency band that rises above whatever this, like you can imagine drawing a curve here for this background, whatever. You have a narrow frequency band in which there is an order of magnitude or more power above and beyond whatever that background stuff is in a narrow frequency band. And you actually see another secondary bump here, which could be a second oscillation. This would be an oscillation in the 7 to 10 hertz range. And this would be maybe a second oscillation of lower power in the 15 hertz-ish range. Or it could be a harmonic of this one oscillation, which we'll cover a little bit later. And so these bumps in the power spectrum are how we traditionally define neural oscillations. So Bu Ha here in a Trends in Cognitive Science paper in 2014 says in a power spectrum, brain oscillations appear as bumps on top of this 1 over f slope. And so in a log power by log frequency presentation of neural data, you have a relatively linear decrease of power as a function of frequency. So power is proportional to 1 over frequency in this log space. And so it's called this background whatever is called the 1 over f. Yuri Bujaki in 2013 in Neuron says when particular oscillation frequencies become dominant, a peak or bump appears in the respective frequency band. So pretty straightforward definition of these oscillations. So what's the problem? It's my favorite question. What's the problem? What are we dealing with here? The problem that we began to run into was that if we study oscillations as bumps, that is a pretty good starting point. I began my career as studying visual attention and working memory in humans. We know from decades and decades of literature that visual cortex in humans is dominated by a alpha oscillation. Alpha is so named because it was the first one observed back in the 1920s by Hans Berger and the first EEGs performed. It's a very clear, very strong oscillation in the 8 to 12 hertz range. And we also know from decades of work that if you present a visual stimulus to somebody, the power in that ongoing alpha oscillation in the visual cortex decreases. So because I am a visual researcher and I'm interested in visual attention and memory, I know that if I run an EEG experiment and I present a stimulus, my power spectrum would go from here in the back to here darker because my alpha has decreased. This blue box here is my 8 to 12 hertz alpha window. This is the window in which I'm looking. And so if I look from pre to post, I have a big change in power, which is what I would expect. What's the problem? This is the model that you have internally, probably that you're working with. If you hear that power decreases as a function of task, or you have a group difference in power between, group A and group B or what have you, this, whether or not you're aware of it, is the implicit model that you're working with. Now, it might be explicit. You might be fully aware that this is actually what's happening to your data, but usually it's an implicit model that you know that if you're going to present a stimulus to somebody and power drops, implicitly this is what you think is happening, whether or not you're aware of it. Now, the problem that we ran into is that we also know, going back to the 1950s at least, that alpha in particular, but pretty much every oscillation as well, can change its central frequency. That is, the frequency can shift. It can speed up or slow down for short periods of time. And so if I, as an alpha researcher, know that I have to look at alpha, which is 8 to 12 hertz, so I filter my data in this 8 to 12 hertz range, and I look at how much power is in that frequency range, I will presume in this analysis that from pre to post stimulus, where the alpha slowed down, and it moved outside of my a priori 8 to 12 hertz analysis window, it will look like alpha power has dropped. So I will see here this big drop in total alpha power. But that is not necessarily the best way to describe what's happening, because alpha power didn't drop. In this case, it's contrived, but alpha power here didn't drop. It just moved outside of my 8 to 12 hertz analysis window. And so, in fact, relative to the background 1 over f like process, alpha power hasn't changed really at all. Total power has apparently changed, but that's not really the best description. Here, the entire power spectrum is also known to shift up or down. So the entire broad frequency range can just shift up or down. And so again, if I'm looking at just my 8 to 12 hertz window here, before and after I present a stimulus, I would say, oh, look, alpha power has dropped. If I filter my data in alpha here from condition A to condition B, it will very clearly show that the amplitude of every individual oscillation cycle is lower. But is that the best way to describe what's happened? I will see a big difference in total power in alpha, in this alpha range between condition A and condition B. But again, relative to that background 1 over f, alpha hasn't changed at all. And finally, another thing that can happen is that the entire power spectrum can rotate around a sort of pivot point in the usually 20 to 40 hertz or so range empirically. We have theories as to why that range is the case, but we've seen this a lot. This goes back to in 2017, we published a paper. Ella Poldvani also published a paper using intracranial visual cortex recordings from patients undergoing surgery for epilepsy. This was with Rafael Malik's group, showing that upon the presentation of a visual stimulus, the entire power spectrum can rotate. And so again, if I'm just looking at my a priori alpha 8 to 12 hertz window, it will very much look like alpha power has dropped from condition A to condition B. So I'll see a big difference in total alpha power, but relative to that background ref process, alpha power hasn't changed at all. And so this is the problem. This is what I got really stuck on, is the way that we often traditionally analyze data by looking at these narrow frequency ranges. Alpha is, of course, 8 to 12 hertz in humans, and theta in humans is 4 to 8 hertz, or maybe 4 to 7 hertz. Maybe alpha is 7 to 13 hertz. But we have these sort of windows of data analysis we want to look at, and all four of these will look the exact same. They will give the exact same result using standard analysis methods. And that's a problem because each one of those four different scenarios may have different physiological interpretations. They might have different physiological generators that are causing those changes, either in a reduction in narrowband oscillation power, a shift in the frequency of an oscillation, a shift in the power spectrum broadband signal up or down, or a slope change of the power spectrum. And we can't adjudicate between those without some kind of careful parametrization. So what's a potential solution? It's one thing to talk about problems. It's better to then start trying to provide some reasonable solutions. And this is where my lab has been for the last couple of years. We have accidentally become a methods development lab for the last couple of years because we cannot get over these problems that we're seeing. And we could talk about them from a theoretical perspective all day long, but the best thing to do is to provide everyone else, ourselves included, with better tools for analyzing and thinking about these things. So the first thing to do is install Python and install some of these software packages. So NeuroDSP is one of the packages that we've created, Neuro Digital Signal Processing. That was initially spearheaded by Scott Cole and Tom Donahue, and then Ryan Hammonds, who is joined my lab as a PhD student in data science, but was the software developer in the lab for the last couple of years, has built out a lot of things. NeuroDSP in this Python package, which is Neuro Digital Signal Processing, initially started as a suite of tools to allow you to do filtering and creation of power spectra using different approaches. Welch's power spectrum, you can do like wavelets or multi-tapers and so on, different ways of estimating your power spectral density. But as we began to do this, we also simultaneously built out what is called the FOOF software package that I'll talk in a minute. This is undergoing a name change. FOOF was originally named because it stands for Fitting Oscillations in 1/F. And this was initially created by a friend of mine, Matar Haller, and the co-senior author on our paper, Avgusta Shestuk, and then built out more in Python by former postdoc in the lab Eric Peterson and then former PhD student Tom Donahue. And so years ago, Matar Haller, Avgusta Shestuk, and I were butting our heads against this problem, and we're trying to figure out solutions. And ultimately, that is what became the FOOF toolbox that Tom built out to what a lot of people are using, hopefully, today. And so the reason that we ended up building NeuroDSP initially began with sets of tools for filtering and creating power spectra. But as we began to build the FOOF toolbox, I'm going to start referring to it as by its new name because it's undergoing this name change to SpecParam. And the reason that we're doing that name change is because we no longer think we're fitting oscillations or 1/F. What we are doing is spectral parametrization. And I may talk a little bit about why we don't think we're doing that fitting of oscillations or 1/F really anymore towards the end of this talk, but also fields other than neuroscience have begun using the spectral parametrization toolbox. I've heard from people in cellular biology, as well as people in sort of the more physical sciences as well, that have begun to use it for domains completely unrelated to neuroscience. But this NeuroDSP toolbox has a suite of simulation tools for simulating I don't want to say biophysically realistic, but a suite of tools for simulating data that have the statistical characteristics that we're looking for. Because if we're going to develop a method to parameterize a power spectrum, for example, then it should work on data that have known parameters. The problem is in biology, we don't have ground truth. We don't know what the real answers are. And so if we're going to create algorithms and tools for people to use that work the way that we want them to, they have to work on data that have known ground truth parameters. And to do that, then we have to simulate data that have very precise features. And so the NeuroDSP toolbox also has a whole suite of functions for simulating different kinds of aperiodic activity, different oscillations that are bursty and non-sinusoidal, and all these things that we'll talk about a little bit later. So let's talk about parametrizing our spectra. Again, like I said, this was started with these dear friends of mine, Matar and Augusta, and then really spearheaded later by Tom, who built out this tremendous toolbox with Eric and some fantastic former undergrads in the lab, who I miss tremendously, Leonda Medanda and Julio Dominguez. And it works like this. So anything that's even remotely related to machine learning could be very, very frustrating sometimes, because this was one of the big bottlenecks that we first faced when we were creating this toolbox, which is I can look at a power spectrum like this, here in black of real data, and I can see that there's this 1 over f kind of curve here. Strictly speaking, 1 over f, the way that people talk about it, shouldn't be a curve, but if you plot it in the semi-log space here, it looks like a curve. And then you've got these bumps atop that curve. And all they wanted the algorithm to do was try and fit that first line. Took a long time to make that work because we wanted this spectral parametrization tools to work on not only non-invasive EEG and MEG in humans, but invasive intracranial recordings, and also local field potential recordings from different animals. So we wanted to make sure this worked on a lot of different kinds of data. And that was time consuming to make it work. But the algorithm begins by fitting this background 1 over f like process and then subtracting it out. So here in B is just that signal in A after you've subtracted out that background fit. This is now our putative oscillations, these bumps above that background 1 over f. And so then what the algorithm does next is it finds the peak in those residuals and tries to fit a Gaussian around that peak. Now, a Gaussian may not be the best, but a Gaussian has been used previously by people who've done peak detection in spectra to try and sort of quantify the center frequency and the amplitude of those peaks. So we began with the Gaussian. Under the hood in SpecParam toolbox, you can actually fiddle around with and swap out fitting functions if you think that they are either better fits and or have better physiological interpretability. But it then does its best to fit a Gaussian, subtracts that out, and then finds the next peak. And it asks if that next peak is above the noise threshold. So you can just do something like z-score these residuals and say, does this peak exceed plus or minus, plus in this case, 2, 3 standard deviations? If so, we're going to still consider it signal and not noise. Do your best to fit a Gaussian to it, subtract that out, find the next peak. That peak is now under the noise threshold, so we can't distinguish that from noise, so we're going to stop fitting. It then says we fit 2 Gaussians, let's fit them both at the same time. Take this dual Gaussian fit in this case, go back to the original signal and subtract this out of the original signal. And that gives you a cleaner background that we can then refit. And this gives us a cleaner aperiodic 1 over f-like background, our 2 Gaussians. We combine them all together, and that gives us then the final estimate of the fit for the power spectral density. And what's neat about this is you can take tens of minutes, hours of data, like from sleep, for example, from EEG, create a power spectral density from that, and a couple of parameters, so this background and 2 Gaussians, explains a huge proportion of the variance of that power spectral density, oftentimes well over 90%. And that's pretty neat, because then you have some way of measuring what the center frequency of your putative oscillations are and the amplitude of those oscillations relative to whatever that background or 1 over f process is. But now we're going to take a little bit of a detour, because there's more to oscillations than just that. The fundamental basis of the Fourier transform is the sine. Sinusoids, adding them together, that gives you a perfect mathematical description of your time series. Now, where's the problem with that? All well and good. One of the problems is I will sometimes hear people say, well, if there is power in the power spectrum of the EEG or LFP, that must mean that there is an oscillation at that frequency in your data, because otherwise, why would there be power at that frequency? In the tutorials that I'm releasing along with this video, we'll step you through some very fundamentals of digital signal processing, including getting the power spectra for white noise data. So white noise, by definition, is uncorrelated with itself in time. There's no oscillations. Every data point is independent from the next. And so white noise, if you look at the power spectrum of pure white noise, it has equal power at all frequencies. That is the Fourier transform of a white noise signal, equal power at all frequencies. So you would be hard-pressed to argue that there is an oscillation at every single frequency in a white noise signal. It's a function of how the math works. If you ask the Fourier transform to return a mathematical representation with a sinusoidal basis, it will give you power at every frequency for white noise. That doesn't mean that there's an oscillation at every frequency. Another extreme is if you have a signal that is nothing but zeros, just all zeros. So no signal except one time point somewhere in there, you have a non-zero element, like a 1. This would be the Kronecker delta, it's called, for discrete time series. If you look at the Fourier transform of that, it also has equal power at all frequencies. The difference between white noise signal and that Kronecker delta, all zeros and a 1, is not in the frequency domain. It's in the power spectra, in the phase spectra. Sorry, not in the power spectra. So the starting phases are all different to make these sine waves all sum up to look like either a single time point that's a non-zero value or uncorrelated white noise. So what's the problem further? The problem is that neural oscillations are non-sinusoidal. These are two examples here. This is just showing motor cortex in patients with Parkinson's disease. This is from a paper by Scott Cole in my lab in Journal of Neuroscience in 2017, looking at the shape of beta oscillations in patients with Parkinson's disease. And what you can see here is you see a very clear beta rhythm, obviously an oscillation. It's a rhythmic process in the motor cortex in these patients, but you see they're sort of shaped like these sharp sawtooth or shark fin shapes. Visual cortical alpha oscillations tend to manifest as these kind of sharp V's. This is a 10 hertz rhythm. These are clearly non-sinusoidal. But if you ask the Fourier transform to give you a representation of a time series that has rhythmic non-sinusoidal oscillations in them, what you're going to get is something that looks like here in the top left. So here is a simulation time series of a non-sinusoidal rhythm at 10 hertz. You can see a very big peak in the power spectrum at the fundamental frequency of 10 hertz. But you also see a peak in 20 hertz. Now, by definition, in this simulation, there is only a single non-sinusoidal 10 hertz rhythm. There isn't a embedded 20 hertz beta oscillation on top of this. This is a single rhythm that is non-sinusoidal. And this is the power spectrum for that. And so in this animation by Natalie, what you're going to see here is we're going to move through different non-sinusoidal shapes here on the bottom right and look at the associated power spectra as we change shapes. This is animated on the bottom left, by the way. As we change the shape of the non-sinusoidal 10 hertz rhythm, you see no difference in the power spectrum. And that's because that change in shape is being captured by the phase spectrum. And so one way to potentially deal with this kind of issue in our data analyses is to look at the phase spectrum. But instead, we chose to sort of adhere a little bit more closely to the data. And so one of the problems we began dealing with when looking at these non-sinusoidal rhythms also was that we can't adjudicate between whether or not you've got a single 10 hertz rhythm and with a second 20 hertz rhythm, or a single non-sinusoidal 10 hertz rhythm. You can't tell the difference. You can't adjudicate between those two without some kind of careful parametrization. And so again, what's the solution? It's one thing to talk about problems. We also like to talk about solutions. And so here, again, the answer is Python. So the NeuroDSP toolbox has a simulation framework also for simulating non-sinusoidal bursty oscillations. And then we have the Bicycle toolbox, which is the cycle by cycle analysis of neural oscillations toolbox, written by Scott Cole and, as I said previously, Tom Donahue, and then in the lab more recently taken up by Natalie, Ryan, and Andrew. and really been sort of building out and leveraging the bicycle toolbox for analyzing the very interesting and myriad shapes that neural oscillations take. And so the idea behind the bicycle toolbox is we've got plenty of data and evidence that suggests that different regions have different oscillation frequencies. So we know, we already talked about visual cortex in humans has this dominant alpha rhythm at 10 hertz. Motor cortex has a beta rhythm. If you look at the hippocampus of mice and rats, that is dominated by a theta rhythm. Now, the theta rhythm in rodents, mice and rats in particular, tends to be faster at like 10 to 12 hertz, which in humans would be the alpha rhythm. But it is a dominant frequency in the hippocampus in rodents. We have gamma oscillations that are higher amplitude, higher frequency, lower amplitude often. We'll see gamma oscillations in visual cortex, for example, and other brain regions. So you have very slow delta oscillations that occur. So we know that we have these different oscillations, and they manifest in different regions. Visual cortical alpha, somatosensory cortex in humans also has a 10 hertz-ish rhythm, but we call it the mu rhythm. So it's not defined by its frequency, it's defined by its shape. It's called the mu rhythm because it has sort of this double M. So not only do we have different parts of the brain that oscillate in different species at different frequencies, but those are relatively stereotyped. And they have these interesting shapes. Hippocampus in rodents is known to be non-sinusoidal. It also has a sawtooth-like or shark fin shape to it. And so what we decided to do, instead of looking at the phase spectrum, we decided to adhere, like I said, more closely to the data. So the bicycle toolbox is a time domain analysis tool where it looks for bursts of oscillations, because oscillations are not stationary. We don't just see a permanent oscillation occur really in anything. Oscillations wax and they wane, they come and they go. Some bursts might last for dozens of cycles. They might last for a very long time, whereas other bursts might only last for 3, 4, 5 cycles. So the bicycle toolbox does its best to try and find these bursts in time of an oscillation. And then once it does that, it breaks each oscillation up into a single cycle from trough, peak to trough. And then it says, okay, for every individual cycle that we have in our data, we are going to measure the amplitude, which is the average of the trough to peak and peak to trough height, the period, which is how long does it take to go from trough to trough in milliseconds, and a couple of really a lot of actual other features, one of which is what's called the rise decay symmetry, which is in how many milliseconds does it take to go from the trough to the peak, and then how many milliseconds does it take to go from the peak back to the trough. For a sine wave, that would be equal. But that's also equal for a triangle wave or a square wave. And so what we also look at is what's the peak trough symmetry, which is how long did the signal spend in the peak relative to how long does it spend approximately in the trough. Now, we don't know if any of these features have physiological relevance or meaning or interpretability, but we can't know if they have physiological meaning unless we first measure them. And so we take the approach and perspective that if you're not measuring it, you can't know if it matters. And so this is an over-parametrization of each cycle. We have how long does it spend in each trough and peak and so on. That's a huge amount of data that's built into, in Python, a pandas data frame that then allows us to over-parameterize each cycle. But once we have these parameters, we can then ask, which parameters are modulated in a disease or a cognitive state or task? Which parameters are correlated with lower level physiological features that we might be able to measure in animal models and experiments. And so we don't know which of these are the most meaningful. And some of these are highly correlated, but by having this rich data frame that it results from every single cycle, we can begin to more carefully analyze which one of these features matters, when, and why. And again, we can't actually know if this is working unless we have some good ground truth simulations. And so the NeuroDSP toolbox allows you to do these sophisticated simulations of bursty non-sinusoidal oscillations, here you can see, embedded in aperiodic background noise. And so you can simulate an aperiodic background with known parameters. You can simulate bursty oscillations that are non-sinusoidal with known parameters, add them together, and then see how well does the bicycle toolbox or other approaches that other labs have developed work on these known ground truth simulations. So we're a big fan of simulating data. When in doubt, simulate is sort of one of the mottos of the lab. And in Scott's paper that we published in 2019 in the Journal of Neurophysiology, sort of making the argument for why this bicycle approach matters, we simulated an event-related design where you have a task that has an onset here at time 0. And at some point in the 1 second after the onset of that stimulus, you have an increase in alpha power. Something happens at some point where an alpha oscillation suddenly emerges. So you have no oscillation in your data, and then an alpha oscillation emerges. And so we simulated, in this case, 3 different scenarios. One is the baseline scenario where you have an alpha oscillation emerge for a couple of cycles. The next was we had the exact same kind of thing happen, except now the amplitude of each individual cycle is higher than in this condition. On average, the burst lasts for the exact same duration. It's just each cycle has a slightly higher amplitude than the baseline condition. Or in a third scenario, we kept the amplitude of every cycle here. Every cycle here has on average the same amplitude as in here. We just had it burst for longer. So the cycles last longer. And if you do traditional analyses, in this case, a narrowband filter in the alpha frequency range followed by a Hilbert transform to give you the instantaneous amplitude across all, we simulated 100 or so trials of this, of each of these conditions. You can see that your instantaneous alpha amplitude is very low up until the onset of the simulated, stimulus, let's say. And you can see that you have, in the baseline condition, an increase in alpha amplitude over the 1 second. But in the condition where we all actually truly increase the amplitude, you do see that over time, the amplitude increases higher than the baseline condition. But in the more burst condition, the average amplitude is even higher. And so it's interesting, right? Because what we're saying here is that traditional methods will say that on average, after the presentation of the stimulus, the amplitude is highest when you have a longer burst. And that is exactly what you would expect the math to do, but that's not necessarily the best way of describing what's happened. It's not that your oscillation amplitude is increased from this scenario, the baseline, to this scenario. It's just that lasted longer, and that's not captured by the Hilbert transform. And so instead, if you use the bicycle toolbox, what you'll see here is that in the bicycle analysis, the average cycle amplitude between the baseline condition in black and the more burst condition isn't different. The bicycle toolbox can adjudicate and differentiate between those two and say, in this case, those aren't different, but we do see in the higher amplitude condition that even though the number of cycles that appear is the same as in the baseline, the amplitude on average for each cycle is higher in that condition. Whereas in contrast, the probability of oscillations increases for all 3 conditions after the onset of the stimulus, but it's higher for the more burst condition than it is for the baseline and higher amplitude conditions. And so rather than just looking at a average event-related change in power or analytic amplitude, I should say, derived from the Hilbert transform of the narrow bandpass signal, instead, using the bicycle toolbox, we can better look at what has changed as a function of our simulated task in this case. And this matters. This isn't just like signal processing fun and playing with numbers. This stuff has some physiological relevance. So earlier on in my career, during my PhD and postdoc days, I was very interested in this idea of cross-frequency phase amplitude coupling. This idea that you can have an ongoing oscillation at a low frequency. Here, this paper, we looked at theta oscillations intracranially from frontal cortex in patients performing these complex cognitive tasks, and looking at the coupling between the low frequency theta rhythm and the power of a high gamma signal. So high gamma is been shown to be strongly correlated. High gamma is like 80 to 150 or whatever hertz. Everybody has sort of different frequency range, but this high gamma, this power in this broad high frequency range is pretty strongly correlated with the underlying population spike rate. And what people have shown And this goes back to like really cool work from Ryan Knolte, from Bob Knight's lab, where I did my PhD also in 2010, in their science paper showing that the power of high gamma fluctuates in time as a function of the phase of, in this case, a theta rhythm. So if you look at the theta oscillation, you'll see that when the theta, say, happens to appear at the peak versus the trough, high gamma amplitude is higher during the peaks than during the troughs. And this is known as cross frequency or phase amplitude coupling, where the amplitude of a faster frequency, in this case, high gamma, is correlated with or coupled to the phase, peak or trough, or anywhere between, of a slower rhythm. And so one of the problems that we began running into in my lab, we like problems, is that for a non-sinusoidal oscillation, this is a simulation here, just showing, very sharp peaked rhythm, that if you filter this sharp peaked rhythm in a beta frequency range of 13 to 30 hertz, it will smooth it out. It does its best. It's just the math doing what you ask of it to fit a oscillation in the beta range to that. So you can see it sort of loses those interesting shapes that you have. And if you filter it in a high frequency range, 50 to 200 hertz, like a high gamma range here, you will see that the high gamma amplitude here in red is highest every time the phase is around 0. This is a cosine phase, so it's toward the peak. So you have very, very clear and strong beta phase to high gamma amplitude cross frequency coupling here. But by definition, the signal that we simulated is just a single non-sinusoidal rhythm. In order for the math, the Fourier transform, to capture these sharp deflections in time, it needs power at higher and higher frequencies. And so if you look at the high gamma amplitude as a function of beta phase, you see it's not uniform. It's very clearly biased, that high gamma amplitude is highest during 0 radians, which is the peak here. And if you look at what's called a comodulogram, which is you look at the phase of slower frequencies here from 5 hertz to 50 hertz, and the amplitude of higher frequencies, 20 to about 200 hertz here, you'll see this very clear preferential coupling between beta frequencies and high gamma amplitude. This looks exactly like cross-frequency coupling. It looks like there's two processes, a beta oscillation coupled to a high gamma oscillation or high gamma broadband signal in a cross-frequency coupling manner. But that's not necessarily the best way of describing what's happened. And Scott really made his point beautifully, I think, in this 2017 paper, looking at this in Parkinson's patients. So he showed that patients with Parkinson's disease have abnormally strong beta oscillations. This is well known. And we showed that those beta oscillations are non-sinusoidal. They have this characteristic sharp sawtooth shape. And so here is a time domain signal in white, and behind it in this colored image here is the spectrogram of that. So it's a sliding window time frequency analysis. So time on the x-axis, frequency on the y-axis. And you can see here for this first cycle of beta, it's pretty smooth. And because it's smooth, the Fourier transform doesn't have to capture any sharp temporal deflections. Like here, the sharp peak. In order for the Fourier transform to capture that sharpness in time, you need more and more power in the high frequencies. And so on this cycle, you don't have a lot of power in the high frequencies, but in this cycle, and this cycle, and this cycle, and this cycle, the sharper they are, the more power you have in the gamma range. That doesn't mean that there is two separate physiological oscillations, a gamma and a beta one, that are coupled to each other. It's just in order for the math of the Fourier transform to capture these sharp features, it needs power in higher frequencies. It's just the way that it works. And so I want to make it very clear, because I find this idea that you have coupled oscillations in the brain super interesting. I love this idea that phase amplitude coupling might be a mechanism by which information could be routed between brain regions. And so when he first started making this argument that the shape of these oscillations matters, and if you just look at something like phase amplitude coupling as a statistical measure, phase amplitude coupling as often measured by most statistical approaches cannot distinguish between a single sharp non-sinusoidal oscillation versus true nested oscillations, where you have a slow oscillation and a faster oscillation embedded in that, whose amplitude increases at particular phases. And so Kareem Zaghlul's lab had this great paper in 2017 led by Alex Vaz, where they looked at a lot of their intracranial human recordings and took every single electrode here, and they've got all these little electrodes that showed significant cross-frequency phase amplitude coupling. And then they went back to the time domain and they said, okay, for every electrode where we found significant cross-frequency coupling, what's happening in the time domain? And they found a large proportion of the electrodes that had significant cross-frequency phase amplitude coupling weren't actually showing two different distinct rhythms interacting with each other. They were just sharp waveforms that looked like coupling between a slow phase and a fast oscillation amplitude. But in some of the electrodes here, you can see in red, they actually do show clear two oscillations interacting. You have a slow rhythm and then a fast rhythm embedded on top of that that has a preferential phase. And so we're not throwing the baby out with the bathwater here. We're not saying that there's no such thing as true phase amplitude coupling between two oscillations. We're saying that you cannot distinguish between those two without better, more careful parametrization, which is what the bicycle toolbox is meant to help with. And to sort of drive this point a little bit home further, Andrew Bender from my group, Natalie Chornikov, are finishing up a paper right now looking at the alpha, visual cortical alpha, and somatosensory mu rhythm. Both have very similar frequencies, very well known to have similar frequencies, but they have different shapes. And so we thought this would be a very good test case to look at the performance of the bicycle toolbox on, in this case, thousands and thousands of people's worth of EEG. This is a very large data set that Andrew's been working with OpenEEG, with thousands of participants worth of data, and looking at using the bicycle toolbox to show that both the alpha and the mu rhythm have rise decay asymmetries that is greater than 0. So they're both asymmetric non-sinusoidal rhythms, but the mu rhythm has more asymmetry to it than the alpha rhythm. So the alpha rhythm, there's sort of these, like I said, deep sharp V's, whereas the mu rhythm tends to manifest as these M shapes. And similarly, the mu rhythm has more peak trough asymmetry, meaning that the peaks happen to be sharper than the troughs, more so than the alpha rhythm, which also has peak trough asymmetry. And this has even been extended into non-neural domain by one of the PhD students in my group, Ina Kosick. Ina has been working with our collaborator at Northwestern University, Christina Zolano, and one of her former PhD students, Torben Nota, who was actually former lab manager, the first lab manager in my lab when it started in 2014. And Christina Zolano studies human respiration and interactions between our breathing and central nervous system oscillations. And so Ina has been sort of taking this approach of, can we quantify the non-sinusoidalities in our respiration and look at how those are related to the non-sinusoidalities in our central nervous system oscillations. And so extending the bicycle toolbox to look at a different physiological signal, like the respiration. So we tend to, our breathing is asymmetric. We breathe in slower than we breathe out on average. Breathing is also weird because we have these long pauses and breaks in breathing and stuff like that. Ina is also extending this bicycle approach to looking at electrocorticography. I mean, not electrocorticography, electrocardiography, so ECG signals, so heart rate, and the different shape of each of the P, QRS, and T complexes in the heart ECG rhythm. So, okay, gonna sort of end here on some 1/f musings. So I've been referring to that background process atop which our oscillation bumps are embedded as a 1/f. That is not a phrase that the lab uses anymore. So what is 1/f really? There's a lot of terms that people use to describe this 1/f background process. And those terms oftentimes carry some historical baggage. So people from different fields have long observed that a lot of real world processes have 1/f-like qualities to them. So if you look at like visual statistics in the real world, visual scenes have a 1/f statistics to them. If you do a power spectrum of anything like in natural visual scenes, they have a 1/f shape to them, meaning that you tend to have, like in my wall back here, you've got this big bare wall, which has a lot of very low frequency content. And then you've got over here, my bookshelf, which has high spatial frequencies in it, but those are lower in amplitude. A lot of processes in physics, avalanches, sand piles, all display these 1 over f-like characteristics. And in fact, depending upon your sort of intellectual background, you will refer to this 1 over f process using different terms. So it might be scale-free or fractal, or it might be indicative of self-organized criticality. Or depending upon where you come from, you might refer to 1 over f-like processes as having long-term memory or long-range temporal correlations. Or you might just refer to it as power law activity or 1 over f noise. But we tend to refer to it now as aperiodic. And why do we do that? Why do we not prefer these terms? First of all, these terms come from somewhere else. They, like I said, carry some intellectual baggage that we don't think is necessarily the best way of describing what we are calling aperiodic activity. So now I'll do my best to defend that. What is the aperiodic signal then? At the very beginning, I talked about what it is that we're measuring in LFP and EEG. So what we're measuring are these postsynaptic and transmembrane currents. By the happenstance and chance of biology, all of these currents tend to have this double exponential shape in the time domain. So for example, a GABAergic inhibitory current will increase or decrease exponentially in time for 40, 50, 60 milliseconds, and then exponentially decay in time over the course of 100, 200, 300 milliseconds or so. That double exponential form here, the Fourier transform of this time domain process is this here in pink at the bottom. So the power spectrum of this is this. In order to capture this sort of sharper deflection in time, you have some power at the higher frequencies, but it's sort of a slower process, so you have a lot of power in lower frequencies. And you can simulate a local field potential as the summation of many of these postsynaptic currents converging on a region all at once. So here is a simulated LFP on the bottom, and this is just the sum of all of these traces on the top. Each one of these traces is just a simulated action potential time series. So we drop we have a bunch of zeros, we include a bunch of ones that are representative of action potentials, and then we convolve each time series, action potential time series, with this synaptic kernel to give you that postsynaptic currents that are being caused by wherever those presynaptic action potentials are coming from. Integrate all of these together, and you get a simulated LFP. Now, in this animation Natalie created, we're going to go from a slower synaptic kernel to a progressively faster kernel. We're keeping everything else the same. The location of each of these spikes here the same. It's just you're going to see that as we convolve these time series with faster kernels, they're going to change their shape, which is going to give a different shape to the LFP, which will also give a different shape to the power spectrum of this LFP. So you can see here, as the synaptic kernel becomes shorter in time, its time scale decreases. It's called the tau is the decay. So initially the tau decay was 100 milliseconds, which is more like a slow GABAergic current. Whereas here, the decay is much faster, closer to like a glutamatergic excitatory current, AMPA, for example. And so you can see everything else here is the same, but the LFP begins to look different. And in order for the Fourier transform to capture the sharpness of this, it has more power at higher frequencies. This form here, the Fourier transform of a double exponential or an exponential, is known as Lorentzian. It has relatively flat power out to characteristic knee frequency, at which point it appears to linearly decay in exponential log space, I should say. And it looks like a 1 over f. So oftentimes when people analyze the 1 over f component of power spectra, they ignore a low frequency range and just fit a high frequency range, because the low frequency range doesn't demonstrate this scale-free property of a linear decay in log space. The reason that we don't think we should be referring to it necessarily as 1 over f or any of those other terms is because, like I said, they carry some intellectual baggage, and we believe that the fact that the power spectra tend to look like this and have this 1 over f-like quality is because the signals that we were recording have a physiological generator that is a double exponential in the time domain that by their very nature have to give this shape in the frequency domain. That is, again, the nature of the math. And so as we move through, to show you again, Natalie's beautiful animation here. And so this argument was put forth by Richard Gao in 2017 in this paper where we were trying to struggle with what is this aperiodic background signal. And I had observed in 2015 in a paper I had published with my former postdoc advisor, Adam Gasley, Journal of Neuroscience paper showing that older adults have flatter power spectra than younger adults. And I'd interpret this as like a neural noise phenomenon with the idea that white noise has a perfectly flat power spectrum. We know that the power spectra of neural data has this negative slope. It's not flat, so it's less pure white noise-like. And older adults had a flatter power spectra, which looked more like white noise. So I interpreted it as noise. And Richard Gao said, I don't think that's right. And so we started digging into it, made him try and prove it, and he did a great job in this paper, showing that you can simulate the LFP as a summation of different excitatory AMPA and inhibitory GABA currents. And if you look at the power spectrum for a purely excitatory signal, it looks just like I showed you in Natalie's GIF earlier. And a purely inhibitory signal has higher power but and lower frequencies, but then drops faster. And real neural data, if you look at the two of them together in the LFP, looks like this. And so as you change the relative contributions of excitatory and inhibitory currents that are contributing to your local field potential, the actual power spectrum that you see will move between these two extremes. And so Richard showed that if you look at the slope of the power spectrum, the exponential fit, we would say, that tracks in simulation the relative balance of excitation and inhibition, totally ignoring the complexity of all of the different kinds of neuromodulatory currents, the fact that NMDA is an excitatory current that has a very long time scale, much longer even than GABA. This is a simplistic model, but it has some nice explanatory power in that it at least explains where the shape of that power spectral density comes from. And so here, again, a great animation Natalie has of simulating a purely excitatory, purely inhibitory current and changing the relative contributions of excitation and inhibition to the local field potential and seeing what happens to the shape of the power spectrum. So you can see here, as you increase the amount of inhibitory current relative to excitation, the power spectrum begins to look more like that initial inhibitory current. And as you decrease the amount of inhibition relative to excitation, the power spectrum begins to look more like the excitatory power spectrum. MJ Preston, Michael Preston in my group with Natalie, we have a paper under review right now showing that the shape of the power spectrum changes dynamically in a task-dependent manner in human visual cortex. And this is a reanalysis of a data set from Fellner is the first author with Simon Hanselmeyer's group, PLOS Biology 2019, I believe it is, with intracranial recordings from human visual cortex. And this is a sliding window analysis of the aperiodic signal, so this background. So you can see here before a stimulus onset, you have this nice aperiodic signal here fit by this yellow dashed line. And then you have an alpha oscillation. Here, it's around 11 hertz, 10 to 11 hertz in this particular patient before the stimulus onset. And then when I play this animation, what you'll see is that the alpha will collapse and disappear entirely, and the entire power spectrum will rotate around a central frequency. Stimulus coming on, alpha disappears, and the power spectrum is rotated. And so you can see just before the stimulus onset versus just after, you get this collapse of alpha and a rotation of the entire power spectrum, where the high frequencies increase simultaneous to a decrease in low frequency power. And this is indicative of this spectral rotation is indicative of a change in this aperiodic exponent. And this happens in an event-related fashion. The exponent pre-stimulus versus post-stimulus, in this case, gets closer to 0, which means it flattens out. And so this is reanalysis of that existing data set by MJ, which really clearly demonstrates that this aperiodic signal is not just background noise, it is dynamic in a functional manner. And the fact that it rotates around this central point here, so what is this? This is 20 hertz right here. So that alpha is probably more like 12 hertz. The rotation point in this case happens to be right around 30 hertz or so. And so that point where those two processes intersect is what we would predict from our aperiodic AMPA excitatory versus GABA inhibitory model, which is kind of neat. It's a simple computational model of the local field potential that makes predictions that bear out in task relevant ways. So like I like to say, this stuff matters. So Sydney Smith, one of the PhD students in my lab, has a paper that's been provisionally accepted looking at this aperiodic activity in major depressive disorder. So Sydney worked pretty closely with these patients with major depressive disorder who are undergoing ECT treatment. Now, electroconvulsive therapy, ECT, has a pretty bad rap. Historically, just the history of psychiatry is not bright, but it is a very effective treatment for reducing depression and suicidality in patients with major depressive disorder for whom other treatments are unsuccessful. So these patients come in, cognitive behavioral therapy, antidepressant drugs are just really not helping them. And so they come into the clinic and they undergo a couple weeks of electroconvulsive therapy treatment, whereby they are given a anesthetic paralytic in an outpatient setting. This anesthetic happens to be, in this case, Bravitol, very brief, short-acting paralytic and anesthetic. And so they're unconscious for a very brief period of time. While they're unconscious, a current is passed across the frontal cortex, large enough to give them a seizure. The target is for the seizure to last around 45 seconds to a minute. They're allowed to recover. The anesthetic wears off. They wake back up. They hang out in the outpatient clinic for a little while. It's they're pretty confused. But even though it's tremendously effective, so here is self-reported depression index, the QIDS, with successive treatments over time for each one of these patients. And you can see over time with each successive ECT treatment, the depression symptoms recover. So it's very effective. And yet, when you talk to psychiatrists and ask, what is the mechanism by which this works? Most don't know. They say, one psychiatrist told me, it's basically rebooting your brain. As a neuroscientist that's interested in physiology and physiological mechanisms that give rise to cognition and treatments, that is unsatisfying. And so Sydney began to run this experiment with our collaborator, psychiatrist here at UCSF or UC San Diego, Mariam Sultani. And Dr. Sultani really helped coordinate this, whereby we recorded EEG just before every fourth treatment and just after every fourth treatment. So before the patients undergo ECT treatment, we record a couple of minutes, up to 10 minutes, hopefully targeted 10 minutes of EEG before the treatment. And then they're given the ECT treatment, have the seizure, and then as they're recovering, we record another 10 minutes after the anesthetic wears off. And what Sydney showed is that from pre to post-treatment, the power spectra, the aperiodic activity in these patients actually increases. It becomes more negatively sloped with each successive treatment. And the degree to which this steepening of the power spectra, this increase in aperiodic activity occurs, is predictive of the improvement in the QIDS, in the depression symptoms. And in this model that we showed you, this is indicative, this steepening of the power spectrum, of a shift towards more inhibition, away from excitation. And so this is an example where understanding the physiological origins of your signal and having a better way of quantifying the data other than just narrow bandpass filters in different frequency ranges gives you a different interpretation of the potential mechanism by which something like ECT could operate. And so that's it. That is the spiel. The take-home message here is that we can think about our data more than a bunch of narrow bandpass filters and a priori frequency ranges. There's more going on in our data than just that. Our oscillations are not sinusoidal. They have rich and interesting shapes that might be telling us something about the underlying circuitry that gives rise to them. This may help us understand better oscillogenesis and what kinds of disruptions and diseases may change the shape of those rhythms or influence the aperiodic background activity, or both. And we can't really know unless we have better tools for analyzing our data. And that is it. I'll post this video and slides along with the tutorials that we've been creating and created for the Society for Psychophysiological Research. And I hope I've convinced you. If not, email me and yell at me at a conference or what have you. And I'll try and convince you in person. All right. Thanks, everybody. Bye.